And welcome back. We're talking about LDL receptors, how they get synthesized, and the fact that there are different types of genetic defects, single nucleotide polymorphisms that can occur that can make somebody's LDL receptor much more effective and efficient than the next person. And you see this when you're looking at people's blood work. You'll sometimes see people who they're thin, their diet's pretty good, they exercise, and yet they have a cholesterol level that is much higher than you would have anticipated. It might be 5.7 or 5.9, or even over 6. And it's like, wow, they don't eat a lot of really bad foods. And very often it's explained by the kinds of defects I'm going to show you right here. So the LDL receptors, when they're synthesized, they're translated by ribosomes on the endoplasmic reticulum. Then they're modified by the Golgi apparatus before traveling in vesicles to the cell surface. So certain things can go wrong along the way, as you'll see. So mutations in the gene encoding for the LDL receptor are known to cause familial hypercholesterolemia. So there's homozygous form, where you just don't make LDL receptors hardly at all, and so you get sky high LDL cholesterol levels and total cholesterol. And then there's heterozygous forms of this, where it's sort of a mix. So you make LDL receptors, but you're not making them as perfectly as the next person might. And so you see these modestly elevated cholesterol levels in someone whose diet and lifestyle is pretty good. And these people have a greater propensity to develop heart disease at a much earlier age, just due to that inborn error of metabolism. But we'll talk about how to work around that. There are 5 broad classes of these LDL receptor mutations. So even though there's 27 single nucleotide polymorphisms, there's 5 major classes of them. So the first one is the mutation affects the synthesis of the receptor in the endoplasmic reticulum. So it becomes difficult to make the LDL receptor in the first place. And so let's look at that for a moment. So if we go to the next slide, I think I put it there for us to look at. So let's go here. So here is the LDL receptor being synthesized in the endoplasmic reticulum. So if this is faulty at this point, then you're not making the LDL receptor very well in the first place, if at all. And then mutations can occur where you prevent the proper transport of that LDL receptor to the Golgi body for some post-translational modification. So this would be the transporting of the LDL receptor to the Golgi apparatus. Sometimes this transport system is defective. And then the third class is its mutations to stop the binding of LDL to the receptor. So the receptor is made in such a way that you make the receptor, but it's not shaped properly so that the LDL doesn't actually fit into the receptor. And then the fourth mutation is the mutations inhibit the internalization of the receptor ligand complex. So this would mean that once LDL fits into the receptor, that the receptor doesn't internalize it and doesn't internalize that vesicle and bring cholesterol into the cell. It just sort of sits there on the cell surface, and you don't really have a chance to complete the functionality of what the receptor is designed to do. Then you can have the receptor can't recycle properly. So what that means is that if you bring LDL into the cell and you release the cholesterol, that the LDL receptor normally recycles back to the cell surface, and then it's available to pick up more LDL again. But the recycling of this LDL receptor, once synthesized, doesn't occur. So you're totally dependent upon just making brand new LDL receptors all the time. So in that case, the person will have LDL receptors on the cell surface, but they won't have as many as would be considered optimal because they're not recycling their LDL receptors in an efficient fashion. So these are the main classes of the mutations, and I really think that all health practitioners who are going to give people advice around diet and cholesterol and saturated fats and exercise should have a working knowledge of this, so that when they look at blood work, they can start to understand where things can go wrong, and why sometimes you see some of these strange elevations in somebody whose diet and lifestyle is actually quite on target. So then we deal with the macrophage scavenger receptor. I touched on this before. Some cells, particularly the phagocytic macrophages, have non-specific receptors known as scavenger receptors that bind various types of molecules, including modified LDL particles that have been oxidized. So modification of LDL frequently involves oxidative damage, particularly of the polyunsaturated fats that are in the LDL. So when you have polyunsaturated fats in your diet, from vegetable oils, so fish oil, flaxseed oil, borage seed oil, that those fats, when they're traveling in those lipoproteins, because they have double bonds, they're very prone to oxidation. Those bonds can break quite easily if they're not protected by, vitamin E and other fat-soluble antioxidants like carotenoids and coenzyme Q10, which can protect those double bonds within the lipoproteins. And if they get oxidized, it becomes problematic. So in contrast to the LDL receptors, the scavenger receptors are not subject to downregulation. So the macrophages just continue to ingest more and more LDL particles until they become totally engorged. So the continued presence of scavenger receptors in the cell membrane allows the cell to take up oxidatively modified LDL long after the intercellular cholesterol levels are elevated. When the macrophages become engorged with lipid, they're called foam cells, something to remember. And the accumulation of these foam cells in the subendothelial space of the blood vessels forms the earliest gross evidence of a developing atherosclerotic plaque known as the fatty streak. So the fatty streak that starts the process of atherosclerosis is formed from cholesterol that gets absorbed by macrophages and become foam cells. The process, and the higher the LDL, the faster this occurs, and the more the LDL becomes oxidized, the faster this process occurs. The processes that cause oxidation of LDL involve the superoxide anion or superoxide radicals, nitric oxide, hydrogen peroxide, and other oxidants. We've talked about how these free radicals get formed in a previous course, Theories of Aging. And antioxidants such as vitamin E, ascorbic acid, vitamin C, and carotenoids like lycopene, lutein, zeaxanthine, beta carotene, the ones that have antioxidant capability are able to also act as an antioxidant inside these lipoproteins to protect those double bonds of the polyunsaturated fats. So when you ingest vitamin E, and when you ingest foods or supplements with beta carotene and other carotenoids, and if you take CoQ10 supplements, that the way that they get transported through the body to the largest degree is that they also, once they get to the liver, are put into these lipoproteins, and that's how they get distributed often to the cells of the body as well. And while they're being transported in the different lipoproteins, the chylomicron, the VLDL, the LDL, they're able to act as antioxidants to protect those polyunsaturated fats. So they're involved in protecting LDL from oxidation, and so this plays a key role. Preventing oxidation of LDL, we think, is very important. They talk about some drugs, including statin drugs, saying one of the ways that it decreases the risk of heart disease is by acting as an antioxidant for LDL cholesterol. So, if statin drugs are given credit for this, then some of these other nutritional antioxidants should probably be given credit as well. A little bit more on the HDL. So let's just review that the HDL picks up cholesterol from the surface of the cells and from other lipoproteins and converts it to cholesterol esters. So these cholesterol esters are ultimately returned to the liver, and thus HDL is said to participate in, and this is the key word, reverse cholesterol transport. So it can take cholesterol out of the cell, the blood vessel wall, but it also picks up some cholesterol from other lipoproteins and also from other tissues that may have excess cholesterol that they need to export. And I'm saying that because in a condition called Tangier's disease, where HDL is not made properly, that the HDL can't pick up cholesterol from these tissues, and cholesterol then builds up in certain organs and tissues and in the eye, and you start to have very significant side effects from that. But the HDL normally transfers the apoproteins ApoC2 and ApoE to chylomicrons and VLDL. We said that. The triglyceride-rich lipoproteins. Once the chylomicrons and the VLDL have ApoC2, it stimulates the degradation of triglycerides of these particles by activating lipoprotein lipase. So it's apoprotein C2 that stimulates lipoprotein lipase to hydrolyze the triglycerides that are in the chylomicrons and the VLDL, so those fats can get picked up by fat cells to form triglycerides inside the fat cell, or picked up by muscle cells for beta oxidation. So two things can go wrong here. What if genetically you don't make apoprotein C2? Then you can't clear triglycerides very well. Or what if you don't make the enzyme lipoprotein lipase? Then even if the ApoC2 is there, you still don't activate the enzyme that will actually clear it. So sometimes you'll see in a blood profile of somebody who's not overweight and they eat a pretty healthy diet, they don't gorge on carbohydrates, and you'll still see these, you might see cholesterol that's reasonably low, but you might see triglycerides that are extremely high. And it's like, how could this be? Well, what could happen is the person doesn't make ApoC2, or they don't make lipoprotein lipase, or they don't make it at the levels that would be required for normal physiological function. But normally this degradation produces chylomicron remnants and the IDL. So as the VLDLs get the triglycerides cleared, they become the intermediate density lipoprotein, and then they move on to the LDL. And the ApoE, which these particles contain, serves as the ligand for receptors on the liver cell membranes that are involved in the uptake of chylomicron remnants and IDL. So as the HDL transfers ApoC2 to the chylomicron and the VLDL, it uses the apoprotein C2 to clear triglycerides. But the HDL also transfers ApoE to these lipoproteins under normal conditions, and the ApoE is the receptor that allows the VLDL and the IDL and the chylomicrons to be cleared by the liver through endocytosis, and by enabling it to bind to those liver receptors. So if there's an ApoE defect, then often you're not clearing these lipoproteins effectively as well. So the nascent HDL particles, so the HDL that's originally made in the liver before anything happens to it in the circulation, they have very little cholesterol ester to start with, and triglycerides. As they pick up cholesterol from other lipoproteins and from cell membranes, and from the atherosclerotic plaque, the cholesterol is converted to cholesterol esters by the LCAT enzyme, which is stimulated by ApoA1. So ApoA1 is another very important apoprotein that needs to be part of the apoprotein structure of the HDL, so that the cholesterol can be converted to these cholesterol esters, which is a component of the nascent HDL particle, ApoA1 is. As the particles fill with cholesterol ester and triglyceride, they become larger, spherical in shape. So the analogy is that when HDLs first leave the liver, they're like a small little kitchenette garbage bag. And then as they fill up with cholesterol along the way, they become this larger, garbage bag that they're basically going to bring that excess cholesterol back to the liver to be degraded. So these HDL particles, as they get filled up now with cholesterol and some triglycerides, are now called the HDL3. In case you're wondering what the HDL3 is, if you ever see that, it's the HDL that actually has picked up a lot of cholesterol and triglycerides in its travels. And it transfers the cholesterol esters to VLDL in exchange for triglycerides. So cholesterol ester transfer apoprotein. So as VLDL is degraded by the lipoprotein lipase, it transfers the ApoC2 apoprotein, originally obtained from the HDL, back to the HDL. So that's interesting, that the HDL has the apoprotein C2, donates it to VLDL for its purposes. Once the VLDL has used it to stimulate lipoprotein lipase, it'll give it back to the HDL. As a result of these transfers of lipids and proteins to HDL and triglyceride degradation, VLDL is converted to smaller and smaller dense intermediate density lipoprotein. So as the VLDL gives up some of its components, especially triglycerides, it becomes the intermediate density lipoprotein. We talked about this. The triglycerides, some of the IDL particles are degraded mainly by hepatic triglyceride lipase. So some of those fats go back to the liver, and ApoE is transferred back to HDL from the IDL. And then LDL is formed from the IDL. So then the remnant particle of the intermediate density lipoprotein then is the LDL. And LDL has a very low content of triglycerides because most of the triglycerides now have been removed, either by fat cells, muscle cells, or picked up again by the liver. But it has this very high concentration of cholesterol esters. And the LDL now lacks apoprotein C2 and apoprotein E. And thus clearance of LDL from the body, from the blood, by the liver and extrahepatic tissue largely depends on the presence on the LDL surface of apoprotein B100. That's the primary apoprotein that needs to be made so that the LDL can be cleared. And if there's a defect in apoprotein B100 synthesis, then it's very easy again for cholesterol to build up in the system. So two of the important ways that high cholesterol occurs from genetic reasons is a defect in the LDL receptor synthesis or transport or recycling, an LDL receptor defect of some kind. Or you don't make apoprotein B100, which is required to clear the LDLs from the system. So certain types of investigation can determine if the person has these types of genetic defects when you see things that are just way off the charts. A person has a fasting total cholesterol of 8.7 or 9.2. You're like, oh, this is not diet. There's an underlying genetic problem here. And just while we're talking about cholesterol, remember that when LDL brings cholesterol back to the liver, that some of that cholesterol can be converted into bile acids. Let's just talk about these bile salts for a moment. The intestinal bacteria deconjugate and dehydroxylate the bile salts once they're secreted into the intestinal lumen, removing the glycine and taurine residues and the hydroxyl groups at position 7. So the bile salts that lack a hydroxyl group at position 7 are called secondary bile salts, like lithocholic acid. A secondary bile salt that has a hydroxyl group only at position 3 is the least soluble bile salt. Its major fate is excretion through the fecal route. But greater than 95% of all the bile salts that are secreted into the lumen of the intestinal tract are resorbed in the ileum and returned to the liver via the enterohepatic circulation. But what if there was a magnet there that could bind to those bile acids and prevent them from being reabsorbed back into the body? Then the liver would have to take more LDL cholesterol out of the bloodstream and convert it into bile acids. So again, that's what soluble fiber does. That's what bile acid sequestrant drugs do. They help to excrete more of these bile acids. The secondary bile salts may be reconjugated in the liver, but they are not rehydroxylated. Because the steroid nucleus cannot be degraded in the body, the excretion of bile salts serves as a major route for removal of the steroid nucleus and thus of cholesterol from the body. So bile acids cannot be converted back into cholesterol. That's the point here. Less than 5% of the bile salts that enter the lumen of the intestinal tract are eventually excreted in the feces. And 95% returns through the enterohepatic circulation. But if you're trying to lower cholesterol, you want to try to excrete more of that bile, so that the liver will be forced to pick up more LDL and convert it into bile acids and excrete that. You keep excreting it over and over again, and your blood cholesterol comes down. Although the fecal excretion of bile acids is relatively low, it's a major means by which the body disposes of the steroid nucleus of cholesterol. So this is the way that the body can get rid of excess cholesterol, is through the fecal route, through the conversion to bile acids. Because the ring structure of cholesterol cannot be degraded in the body, it's excreted mainly in the bile as free cholesterol and bile salts. Okay, let's move to atherosclerosis, so we understand all the steps involved. So we have this decreasing vessel elasticity. The plaques that you see in the artery wall in atherosclerosis are composed of smooth muscle cells, connective tissue, lipids, and debris that accumulate in the intima of the arterial wall. So the endothelial cells of the artery wall are injured. This is the first thing that happens. You get denuding of the endothelial lining. This can be done mechanically by a lot of turbulence in the bloodstream, or by cytotoxic agents, including oxidized LDL cholesterol can actually damage the endothelial lining, and homocysteine can also do this. The injured area is exposed to the blood and it attracts monocytes. So just like if you were to cut yourself, monocytes would come to the area, and then they would morph into macrophages that engulf the material in the region. As these phagocytic cells fill with lipid, because they have these scavenger receptors that start picking up LDL, they become foam cells which accumulate, causing a fatty streak to develop within the blood vessel wall. The endothelial cells normally produce prostaglandin series 12, which is a prostacyclin that inhibits platelet aggregation. So endothelial cells normally are preventing a thrombus from forming by making sure the platelets don't clump together abnormally. When endothelial cells are damaged, as occurs in atherosclerosis, the platelets begin to aggregate and they release thromboxane A2, which makes them stick together, a substance that further stimulates platelet aggregation. These cells also release platelet-derived growth factor, which causes the connective tissue in the smooth muscle to start proliferating, growing into the lumen and the subendothelial space. And macrophages produce additional growth factors. The growth factors cause proliferation of smooth muscles, which migrate from the medial to the intimal layer of the artery wall. Cells within the intimal layer release lipids, which accumulate in the developing plaque, and blood lipoproteins, particularly LDL, continue to enter the area, contributing to lipid buildup. Cells in the lesion secrete collagen, elastin, and glycosaminoglycans, forming a fibrous cap, and cholesterol crystals appear in the core of the plaque. So the cells are trapped now in the plaque where they die, adding to the debris, and then over time the plaque gets calcified on its most external surface. Rupture and that makes it very rigid. So then rupture and hemorrhage of the encapsulated plaque in a coronary vessel may cause the acute formation of a clot, a thrombus, which further occludes the vessel, causing a myocardial infarction. So these are sort of all the steps that occur in atherosclerosis leading to a heart attack or a stroke. So this is a nice little diagram that sort of shows a lot of these events. And so you can see the internal lamina. You're seeing here's oxidized LDL cholesterol, and you see a scavenger receptor that's on the macrophage picking up the L. Here's a macrophage, and here's a scavenger receptor picking up the LDL, and you see smooth muscle proliferation. And you have the formation of these foam cells as the LDL accumulates to a greater and greater degree over time. So I just thought I'd show you that because it takes us to this overview, this sort of final overview of lipoprotein physiology. Because these lipoproteins are so intimately involved in atherosclerosis, I found this particular illustration that I think is really excellent. It sort of ties all the pieces together. So when you eat dietary fat and cholesterol, in the intestinal cells, it forms chylomicrons. And then the chylomicrons in their travels, once they get into the bloodstream, after they've been absorbed through the lymphatic system, when they come into contact with the endothelial cells around the fat tissue and muscle cells, with the help of apoprotein C2, which they get donated from HDL, it activates lipoprotein lipase, and then free fatty acids can be delivered to fat cells and to muscle cells. The adipose tissue cells will store that as triglycerides and possibly release that at a later point into the bloodstream as free fatty acids and glycerol at some point down the road. And then the chylomicron remnants that are still remaining will get picked up by the liver, and it'll require ApoE in order for that receptor to be stimulated. And the ApoE again was donated to the chylomicron by the HDL. That's where the chylomicron got it. And then once you have fat and cholesterol in the liver then secretes the very low density lipoprotein, and once again the VLDL picks up apoprotein E and apoprotein C2 from the HDL, and with the apoprotein C2 you can stimulate lipoprotein lipase, and fats can then go into the fat cell and to muscle tissues for use as beta oxidation to generate energy here. And then as the fats basically get picked up, you form the intermediate density lipoprotein, and some of the IDL can actually go back to the liver, and with the apoprotein E it can get picked up, and the fats and remaining cholesterol and protein can be reutilized for other purposes. But then a lot of the IDL, as the triglycerides get removed, get converted into the low density lipoprotein, the LDL. And LDL, then, where there are LDL receptors on extrahepatic tissue, the LDL can get picked up and deliver cholesterol to those tissues, which they will use to make their cell membrane. And remember that the adrenal glands and the testes and the ovaries will take that cholesterol and convert it into the steroidal hormones that it needs. In the skin, that cholesterol can become vitamin D. And of course, in order to clear that LDL from the system, you need on its surface apoprotein B100. And the LDL that's not cleared completely by all the extrahepatic tissue, some of the LDL can actually be cleared by the liver cells as well, provided it has apoprotein B100 on its surface to fit into the LDL receptor. And once that cholesterol going back to the liver, some of that cholesterol can be converted into bile acids and secreted into the lumen of the artery as bile acids here. And normally 95% of the bile acids get reabsorbed and come back to the liver. But if you could, in the intestinal tract, sequester them so that they leave the body through the fecal route, then it forces the liver to pick up more and more LDL cholesterol, convert into bile acids, and it leaves the body through the fecal route, and that helps to lower the total circulating LDL cholesterol level. And that's why soluble fiber is such an important way to keep LDL cholesterol down, because soluble fiber from beans and peas, including soybeans and soy products, and from oat bran and ground flaxseed and psyllium husk fiber and oat bran. We've covered these before, but these are really important agents to be. This is medicine. It's not just food. This actually has a tremendous medicinal effect on keeping cholesterol in the ideal range. So a summary of atherosclerosis, you get LDL oxidation, foam cells are formed, we talked about that, the recruitment and proliferation of smooth muscles, and the synthesis of connective tissue proteins as well, and then the formation and organization of arterial thrombi. So these are sort of the events that occur in a short summary. And here it is with some illustration. First you have an injury, the denuding of the endothelial cells, lipoproteins collect in the lining of the artery, macrophages go to the site as part of the inflammatory process, platelets, cholesterol in the blood stimulates smooth muscle cells and connective tissue proliferation abnormally. You get the yellow fatty streak on the inner lining of the artery where the fibrous plaque develops. Collagen fibers proliferate and blood lipids accumulate, which include the blood vessel lumen. So you just see the series of events here, including coronary spasm. And so I think we'll leave it there. When we come back, we'll just have a closer look at the lipoproteins and the different apoproteins that are on their cell surface.